Thank you everyone for joining today's webinar. We will shortly start our webinar. We just have 4 minutes to start our webinar, and we are trying our best that we will start our today's webinar on time. But it is request for all the participants, please mute your mic during the complete webinar, and after the question where in Q&A session you guys may ask the questions by raising the hand. I think am I audible to everyone? तो प्लीज अगर कुछ प्रॉब्लम है तो कोई चैट के अंदर रिस्पॉन्स कर सकते हैं. यू गाइस कैन इजीली रिस्पॉन्ड इन चैट. Ronald, what is the time at your end?
Now is 12:57
Okay,
a.m.
means
p.m. now.
Your lunch time is very close.
Yes, already finished.
Oh, that's great. That's great.
Yeah. So could you explain who is the participant for this seminar? This is academician and also from the sponsor side or the pharma industry?
A group of people, some are from academics, some are from pharmaceutical industry, and some are from the sponsors like other B centers.
So majority is also from the Pakistan?
Some are from Pakistan, some are from other countries, but we are expecting majority is from Pakistan.
Okay, okay.
I think it's 11. Now, Ronald, we should start.
Okay, sure.
Okay.
Please.
Bismillahirrahmanirrahim. Assalamu alaikum. A warm welcome to you all. Today's session. First of all, I'm very thankful to our respectable guest, Ronal Simanjuntak. He is from Equilab and his education. Am I audible to everyone?
Yes.
Right? Okay, perfectly. If I talk about his academic career, he is Masters in Management and Apothecary and also the Bachelor of Pharmacy. Ronal Simanjuntak has over 15 years of experience in contract research organization with a strong focus on clinical trials, bioequivalence studies, pharmacokinetic studies. He has managed over 1,500 clinical trial projects across a wide range of therapeutic areas, including bioequivalence and pharmacokinetic studies for global regulatory submission as well. Cosmetic claim testing and other laboratory testing. He brings expertise in clinical area, regulatory project management, and business development. Currently, as the president, Ronald continues to lead the company's growth, reinforcing its position as a leading contract research organization in Southeast Asia, which is Equilab International. So we warmly welcome Ronald Simanjuntak to today's webinar, and today he is going to discuss technical aspects of the bioequivalence studies. And I'm also very thankful to all the participants who joined today's webinar. And it is a great opportunity for all the Pakistani pharmaceutical industry people to get the idea of the bioequivalence studies, to learn and to ask their questions from Simanjuntak and also from Dow University of Health Sciences. Basically, Equilab and Dow University of Health Sciences are in close coordination for the conduct of the bioequivalence studies and so many projects. So without any delay, I request Ronald Simanjuntak to please continue today's session and deliver knowledge with you guys. Thank you.
Yeah, thank you very much, Dr.
Ronald.
Syed.
Go ahead.
Yeah, thank you very much for the welcome introduction and also information about my biography. So anybody can share screen, please? Could you share the slide?
Please give me permission to share the screen.
We can also share the slides, Ronald,
yes,
if you
please,
guys
please,
want.
Please share, Dr. Syed.
Okay. I think Wahab has make you guys a host, so you guys can share the screen. Yumna and Wahab, can we share the screen? Because I am unable to share the screen.
As I mean, check the back sizes of the Luciana 20. I'm just sharing you. I will be sharing. I will be sharing you after the kind of community. इसके book the equals of the meeting day. So we have checked this thing previous 3 months in back. What is this? SK no, meeting. There is a
I will manage. Yeah, please. Too noisy.
Yeah, okay.
Yeah.
Okay. Asma, Eric, please close your mic. Asma, Eric, please mute your mic.
Close your mic.
Tiffany belum bisa share screen.
Gak bisa, Pak. Aku nggak dikasih akses nih.
I cannot share screen.
Okay, no worries. We can share. We will share. Just give us 5 minutes.
Yes, please share screen.
Yeah, we can share. Meanwhile, Ronald, you can introduce your organization and about you. If I just give the overview about you, but you can explain some other aspects of you and your organization here without.
Yeah, okay. While we are waiting for the share screen, yeah, let's introduce our organization, Equilab International. We are the first CRO in Indonesia and Southeast Asia that already accredited by the WHO and also the 𝐂𝐬𝐻RA. Currently, as mentioned by Dr. Syed, we have managed more than 1,500 projects. This is including for the bioequivalence study, clinical trials, and also the dermatology testing. So for today, I will present about the technical aspect in bioequivalence and bioavailability study. So thank you very much for the DOE University of Health Science Pakistan to give us the opportunity to present about this topic. Could you also make it in the full screen, please?
Yeah.
Yeah,
please check.
Yeah.
Go ahead, Ronald. You can continue.
So during our presentation, I will present about all the related to the study design of the bioequivalence study, and also related to the guidelines and related to the correlation between in vitro and in vivo also. Next slide, please.
Yeah, I have changed the slide. It is visible at your end?
No. How about the other participant? The slide is already changed.
No, it's not visible. It's not changing, and it's not in the full screen mode.
Could you make it in the full screen? Yeah.
Okay, wait.
Yeah, so to continue about the Equilab experience, yeah, we also having experience conducting many study for the innovative drugs. So currently, as we know, many of the development in the innovative drugs, we will share to you also in this slide. So, okay, can you back to the previous slide? Okay, so this is about our agenda. Today we will present about the introduction about bioequivalence study, because we noticed some of these participants is coming from the university and also the academician. So we will explain a little bit about the introduction about bioequivalence. Number 2 for the study guideline. Number 3 about the study process. So some of the pharma company and also from the sponsor side and the CRO side also joining in this seminar. So number 3 and number 4 is covered for the CRO part. Number 5, this is very, very important for the sponsor side because you want to know about the in vitro and in vivo correlation. And the last, we will go for the Q&A session. Okay. Next slide. Yeah, so about the bioavailability, if we talk about the bioavailability, there is two critical points that we need to assess. The first is related to the rate and the second is related to the extent. Rate of the active ingredient that absorb in the body, and the second, this is about extent, and we translate it in our pharmacokinetic as the Cmax.
The slides are not visible.
For me, it's visible now. For the rate, this is AUC, and the extent, this is for the Cmax. And we call the bioequivalence.
Slides are not visible.
If two products are called bioequivalent
at your end,
similar bioavailability.
It is visible with, to the other people. Yeah, please continue. Ronald, you can continue.
Two products called bioequivalence if they have similar bioavailability, meaning that similar is a UCT and the Cmax, they have the similar part, and also in terms of the statistical calculation, they have the similar calculation. We will see in the next slide about the criteria of the bioequivalence. Okay, next slide. Next slide, please.
Yeah, change it. Is it changing at your end?
No.
I think there is an internet issue.
Otherwise, I will share my screen. Wait.
Yeah, you please try to share from your end. Maybe it will be easy for you.
Can you see now?
Yeah, we can see. Yes, yes.
Okay. So the purpose of the bioequivalence study, the first thing is related to the therapeutic equivalence. We need to assess the equivalence about the product without doing the clinical trials. As we know, for the new drug, we need to do and we need to conduct the phase 1, phase 2, and phase 3 clinical trials. But for the bioequivalence study, we only conduct this one study only to assess the equivalency in terms of the Cmax and also the AUCT. And also this is to assess the therapeutic equivalence. And this is the model of dosage form performance. When we doing the bioequivalence study, the dosage form that we consume, it will be changes to the drug in solution. And also the will enter to the gut wall and will be entered to the blood. So in pharmacokinetic study, in bioequivalence study, we measure the pharmacokinetic level of the drugs in our blood without doing the clinical PD or the clinical or PD measurement. For clinical study or the phase 1, phase 2 or clinical trials, normally we doing the therapeutic effect to see the clinical equivalence. But for the bioequivalence study, we only doing the pharmacokinetic measurement to check the blood, the assay in the blood to see the equivalence. Okay, the next slide, this is for the bioequivalence evaluation. So there is two that we assess. The first is related to the Cmax, is the concentration maximum in the blood, and the AUC is the area under curve. Area under the curve to explain or to translate the total of the drug concentration in our body during that period, during the certain period. So this is the certain period. And the example of the pharmacokinetics of the bioequivalence study, we also can see in the right side, we compare two drugs, test drug and reference drug. This is the example of the profile of pharmacokinetic. We compare two drugs until, for the example, until 144 hours. But normally for the bioequivalence study right now, we perform or we can truncate it until 72 hours. General acceptance for bioequivalence criteria, there is two. AUC with the acceptance criteria 0, 80.00 and 125.00%. And for CMAX also same, the parameters, the same criteria, 80.00 until 125.00%. Different with the drug with the narrow margin safety, we should have the narrow margin. So the parameters in terms of the statistical and confidence interval will be 90%, 90.00 until 111.00%. So some of the examples of this narrow margin like the digoxin, and also currently the new drug like the divalproic acid also should follow this range. And for highly variable drugs, we can widen the CMAX criteria. We will explain later on in the next slide about what is the highly variable drugs, what is the example of the highly variable drugs. Okay. So the next, this is about the guideline in terms of the bioequivalence study. For the BABE study, general guideline that we use is from the EMA and also from the FDA guidance. From the EMA guidance, we can use the EMA guidance in January 2010. This is the common guidance that we use for the whole process in the bioequivalence study. The other guideline that we still use and also still valid until now is like for the FDA guidance in 2003, and also the some of the guidance from the WHO, including Annex 9. This is 2006. Annex 9 for the technical series 1003. And the last one that we also use from the US FDA, this is in December 2022. This is about the statistical approach to establish the bioequivalence criteria. Specifically to the GCP and GLP, because in the bioequivalence study there is two big parts. The first is for the GCP, the second is for the GLP. GCP we call it for the clinical aspect, and the GLP, this is for the laboratory aspect or the bioanalytical aspect in the bioequivalence study. For the GCP, the newest one for the GCP guidance is for the ICH E6 revision 3 January 2025, and also some of the previous EMA guidance specifically to the method development, and also to the this is specific for the clinical part from the EMA. And for the number C in terms of the GLP guideline, bioanalytical aspect, the newest one that we use for the bioanalytical aspect is from the US FDA in 2022. And also one of the guidance for the bioanalytical validation and sample analysis in 2023. So this is the common guidance that we use for the bioequivalence study, because we should follow all of these guidance during our conducting the bioequivalence study, including our SOP. Our SOP also should capture all the requirement from all these guidelines. Okay. Moving forward to our standard process for conducting the bioequivalence study in Equilab. So this is the standard that we perform, like for if any request from the sponsor or from the other university and or for the CRO company, we should do the feasibility. Feasibility purpose to review the study can be conducted in our facility or cannot. Some of the projects also we cannot do in our site because due to some of the limitation, especially in terms of the clinical and also the laboratory part. But if the study, we can conduct the study in Equilab, then we can make the study proposal, and then we can, if the sponsor agree with our proposal, we can proceed for the study signing agreement. And if the method is not yet validated in Equilab, we can do the validation of the methodology test. But if the method we already validated, that we do not need this part, we can directly to make the protocol and send the draft protocol to the sponsor. Currently in Equilab, we have already validated more than 300 molecules. So if the molecules is already validated, no need to do this part. We can directly to make the draft protocol and send the draft protocol to the sponsor for sponsor review. If the sponsor already confirmed for the final protocol, and then we can submit the final protocol to 2 institutions in Indonesia. For Indonesian regulation, we should submit the protocol to our ethics committee and also to our local FDA. In Indonesia, we call it the BPOM for the local FDA. And after we got the approval from the local FDA in Indonesia, we do the study. We can start the study and doing the volunteer screening. What is the parameter that we need to check during the screening? This is the physical and the laboratory examination. And after we got the result for the physical and laboratory examination, then we can continue to the volunteer selection. The volunteer selection based on the study protocol, the number should be based on the study protocol. And we will go for the drug administration, and we'll explain to you later about the drug administration and also related to the randomization. And after we give the drug administration, we will continue for the sample collection and the preparations. So this is the sample collection and preparation. Also, we take the blood sample based on the sampling point. And sponsor can do the monitoring during the sample, during the blood sample. And the sample we will store and manage in Equilab before we doing the sample analysis. Sample storage we can store in -20 or -70 or -80 based on the validity that method validation and also the stability sample that we have performed previously. After we doing the sample analysis, we using our instrument. Instrument can be LC-MS or can be HPLC based on the validation method that we perform. We continue for the data management and doing the code breaking. Code breaking is we open the code because the study is under randomization. So we don't know who is the subject that received test drug first or who is the subject that received reference drug first. We don't know. So during the code breaking, we can open the code and we will put the code one by one subject and doing the data processing and statistical analysis. And after that, we can do the final report finalization. So moving to the study design, as mentioned, the common study design for the bioequivalence study, this is the 2-sequence, 2-period crossover design with the washout period. So the subjects that we doing the screening, we doing the randomization. So the subjects didn't know who is the receive the test drug first or reference drug first. The sequence can be like 2 sequence here. Subject sequence 1 is the T is the test drug, R is the reference drug. Sequence 1 will be TR and sequence 2 will be RT, reference first and then test drug. So this is like the tabulation of the subjects. All of the subjects we will tabulate and we doing the randomization. In Equilab, we use the randomization based on the Dixon and Massey randomization number table. And between the period 1 and period 2, we have the washout period. Washout period meaning that to clean the concentration of the drug from the phase 1 not carry over to the period 2. So this is the design, and the design of the washout period should be 5 times multiplied to elimination half-life. So it should be captured in the washout period about the elimination half-life. Moving to the sample size, because this is also very, very important in clinical study, in the bioequivalence study, we should calculate the number of subjects based on the CV intra-subjects. The CV intra-subjects, how we got the CV intra-subject? CV intra-subjects we can got based on our previous experience or based on the study literatures. So if someone asking about we don't have experience, we don't have the study literature, we can do it like the pilot study first about for this molecule. But if we already have the published literatures about the percent CV, percent intra-subject coefficient variation, that we can determine the sample size. For the example here, we give the CV intra-subject, if we got 15, then the sample size minimum, this is the minimum, is 12 subjects. If we got the CV intra-subject based on literature is 30, then the sample size minimum should be 40 subjects. Yeah, so this is the common sample size calculation for our study we use for the crossover design. But how if the molecule is the highly variable drugs? Highly variable drugs, we can see recently many molecules like categorized as the highly variable drugs. Highly variable drugs is the drugs with intra-subject variability is more than 30% in terms of the pharmacokinetics parameter Cmax and also AUCT. What is the meaning of the intra-subject CV more than 30%? Can somebody help me to mute? Please? Yeah. Okay, so this is the normal variability. It should be like the green one. The range of the confidence interval is 80% until 125, as I explained before. If the variation is low, can be like this, the blue one, and the result is still pass, even though like the coefficient the confidence interval is like 85 to 100 or 85 to 95 is still pass. The bioequivalent is still good. But if the molecule is categorized as the highly variable drugs, the red one we can see here, the variation of the subject is very, very high. And if not meet the acceptance criteria 80 to 125, it will
Excuse
be
me,
paid.
sorry, your slides are not moving. We are stuck on the study design slide.
Already moving now?
No.
Oh. Anybody also can check?
No, Ronald. Slides are not.
Okay,
Yeah, the same. Slide is
Not
stuck.
moving.
Wait, yeah. Not yet
Not
moving
yet.
now?
Change yet.
You have to change your slide.
Oh, coba aku share screen, Kak. Loh.
Hello, has the session ended?
No, please wait. We are having a technical issue.
No, it's not ended.
No, it is not ended. Hello, can you hear my voice?
It's not
Hello?
ended.
Yes.
Yes. Okay, please wait. Yeah, we are reconnecting.
No, session is ongoing. There is just a slight technical issue. I think we are resharing the screen so that everyone can check at their end. Please check. Are the slides are viewable for everyone?
Can you see my screen?
Yes, they are visible.
Yeah. Okay.
Is it working?
Please go ahead, Ronald. Now you can carry on. I think sometime it hangs, na, because of the internet issues. You can carry on from the slide that on which you are.
So anybody already see the slides about the HPD for the highly variable drugs?
No. Ronald, at our end,
No.
it's a slide 1 is visible to us.
Okay, wait, yeah.
Yeah. Please put it on screen mode. Also try to change the slide for checking. Yeah, okay, perfectly fine. Go ahead, carry on. It's okay, Ronald. Now you can carry on. Ronald, you are on mute. Please unmute yourself.
Gimana sih lu Tiffany? Jelas. It's clear now?
Yeah, perfectly clear.
Okay, yeah. So explain about the highly variable drugs, normal variability, low variability, and highly variable drugs. Highly variable drugs, the confidence interval can be from outside of the 80 to 125. So this is the cause of the highly variable. Why the product, some of the product is under highly variable drugs? Because some of the drug substance like statins group, this is also the highly variable drugs. And also some of the product is in term of the formulation and also the dosage form and the release, it will be also impact to the highly variable drugs, and also related to the administration condition. For the example, interaction with the food, and also related to the physiological factor like the hepatic metabolism, and also related to the polymorphism. Polymorphism also can impact to the highly variable drugs. What is the solution and approach to achieve BE result? The first is we can use the reference average bioequivalence. This is for the statistical calculation, and also widening the BE acceptance limit. How to widening BE acceptance limit? We can use the replicate study design. Replicate study design, and also we can use
We can't see the screen. It's not visible.
Sorry for the inconvenience. Ronald will be back in a few seconds. I think there is an internet issue. If any, I think there is an internet issue at your end. I think please check.
अब मुफ्ती साहब ये गुना या तमगा जो भी है वो अपने पूरी दुनिया को अपनी लपेट में ले लिया। इस तरमीम की मुखालफत करने वालों से उनका कहना ये था कि 78 सालों में पाकिस्तान को ये मकाम नहीं मिला जब
Hello,
आपके सारे
sorry,
वज़रा अज़रबैजान में खड़े हैं। फिर
I don't know.
बढ़कर और
I don't know. Somebody remove the notification. Somebody remove me from the Teams. Can we continue?
Yes, please.
Yes. Sorry, somebody remove me from the Teams. We can continue. Hope somebody also can help me to control the others. Okay, so examples of the highly variable drugs, the statin, the intra-subject CV range 30 to 40%, and some of the PPI like lansoprazole, omeprazole also have the intra-subject CV 30 to 40%. Antifungal like the intraconazole, ketoconazole also 30 to 40%, and other classes like the progesterone, natrium diclofenac, and mesalazine also have the intra-subject CV is higher, is 30 to 40%. So for the sample size of the highly variable drugs, we can use the method with the replicate design. Replicate design, we can determine the number of subjects based on the intra-subject CV. For the example, if the intra-subject CV is 35%, if we use the EMA guidance, the number of subjects minimum completed should be 29. So sometimes we propose if the number of subjects is 35%, the number of subjects join the study can be like 33 or 36 subjects. And if for the full replicate design, normally if the intra-subject CV is more than 50%, we can use the full replicate design, four-way replicate design. And the number of subjects required for the example for 50%, it can be 22 subjects completed the study. So for the inclusion criteria, sometimes we use 26 subjects. Okay, so for next to the study procedure, as explained for the blood sampling, we can use the blood sampling criteria based on the guidance. The sampling times minimum 12 times, but currently I think in the average, the number of sampling times, it can be around 19 or 18 or even 20 sampling points. But the guidance mentioned that the sampling times minimum 12 time points, one time at time 0, 2 until 3 before Cmax, 4 until 6 around Cmax, and 5 until 8 after the Cmax until at least 3 times of the elimination half-life. Washout period is minimum 5 times elimination half-life, and the bioanalytical that commonly we use for the bioequivalence study, we can use HPLC, UPLC, LCMS-MS, and LCMS-MS. And based on the guidance, the limit of quantification is minimum 1 per 20 Cmax. Okay, so the intra-subject coefficient variation, this is more deeply about the explanation. Yeah, so this is very important to determine of the number of subjects for the bioequivalence study. The definition of the intra-subject coefficient variation is the variation within the same individual, meaning that if I consume the product today and if I consume the product next week, that will be variation between me. After I consume today and after I consume next week, it will be have a variation. This is the simple definition of the intra-subject coefficient variation. And this impact to the study design, if the coefficient variation is lower than the simple study design like the crossover we can use, but if the coefficient variation is high, then we can use the partial or fully replicate design based on the intra-subject CV that I explained the previous. And And this is the graphic between the intra-subject CV and approximate of the sample size. We have case study here compared to the pilot study and the pivotal study. For the example, for this case study, the intra-subject CV is for the AUCT is 4.16%, and the Cmax is 21.9. Okay, and this is the example of the case study of the pilot study. And if we doing this pilot with 12 subjects and we doing the simulation with 26 subjects, then the result is quite similar. The result is quite similar.
HDP तो ये bottle है जो हम use करते हैं, वो नहीं है ये material.
So the pivotal,
Adnan sahab, please mute yourself.
HDP है ऐसे कुछ.
The pivotal will be 22 subjects. Yeah, the pivotal will be 22 subjects, and the number of the intra-subject CV we got in actual is 5.41% for the AUCT, and the for Cmax 22.87%. So commonly, the product is like this: is the BCS class 1 and the BCS class 3. We will explain you in the next slide about the BCS classification. And if you are asking, do we need to do the pilot study for this kind of the molecule? From my opinion, is no need to conduct the pilot study. We can do the pivotal study directly for this kind of the molecules. Okay, and next, this is the case study 2. We compare how the pilot versus the pivotal study. During the pilot study, we conduct the crossover study, and we see the result. For the intra-subject CV of AUCT is 21.75%. The Cmax is 46.83%. Yeah, and the range of the Cmax, we can see the red one here is 63 until to 128. So we can categorize this molecule is the highly variable drugs. Why? Because we can see from the Cmax, the intra-subject CV is more than 30%. And the result is also not good. Meaning that this product or this formulation is not good, or the study design is not good. We can see based on the intra-subject CV and also based on the GMR. We perform the simulation to see this molecule is potentially bioequivalent or not. So we're doing the replicate design with 33 subjects and 66 subjects. This is the simulation part. If we're doing the 33 subjects with the replicate design, the Cmax is still lower. We can see here the Cmax is still lower, 76% until 107%. And if we're doing the very big number of subjects with 66, the result is good. Last time we conduct the study with we decided with the sponsor to doing the replicate design with 30 subjects. This is replicate design. And for replicate design, as we mentioned, the acceptance criteria we can widen, not 80 to 125, but we can widen for the 30 subjects with the intra-subject CV 37, we can widen to 74 until 134%. So this study or this molecule, we can conclude as the bioequivalence because the range of the confidence interval here is 82 until 17 until 109.07 is already meeting the acceptance criteria here. So this is the conclusion of this product. The conclusion is the bioequivalent. Even though we show in the pilot study this like, oh, this is not really good because the range is very, very high. The intra-subject is very, very high. But when we doing the simulation and we change the study design to the replicate design, the result can be meet the acceptance criteria. But this is the replicate design and we can widen the criteria. Hopefully it is clear for this replicate design. Okay, next we are moving to the BCS class. BCS class is also very important from this. from the sponsor side, because you are developing the product, and also when you develop the product, you need to know your product, including the BCS class. BCS class is the scientific framework divided into 4 groups according to the solubility and the permeability. So there are 2 points in this BCS class: the solubility and the permeability. High soluble, we can say that the molecule is the high soluble if the highest dose, this is the highest dose, insoluble in 250 milliliter or less in all of the pH. So the definition of the high soluble in the BCS class is not really same with the high soluble in your practical based on the USP pharmacopy, but this is the highest dose should be soluble in 250 milliliter or less in all pH. And highly permeable drugs, we can say the molecule is the highly permeable drugs if the product have 80% or more in terms of the bioavailability. So meaning that 80% can be absorbed in our gastrointestinal tract. So this is the example of the BCS class 1, 2, 3, and 4. BCS class 1 is the high solubility and high permeability. The example of this molecule is like the amlodipine and others. For the BCS class 2, this is low solubility, but they have a high permeability. BCS class 3 is the high solubility but low permeability, and BCS class 4 is low solubility and low permeability. We can see all of the example of each BCS class here. And we need to know, and we should know the BCS class of our product. So this is important to determine our strategy in terms of the development and also in terms of the prediction to get the bioequivalence study result. For BCS class 1, the absorption of the rate control is based on the gastric emptying. So normally the BCS class 1, the result is the bioequivalent. Not really a challenge to get the result. This is commonly, if the dissolution is good, then the bioequivalence study is also good. Some of the molecules also already biowaver, and WHO also already give some recommendation for some molecules to do the biowaver for the BCS class 1. So no need to conduct the bioequivalence study for some of the BCS class 1. For BCS class 2, the absorption rate control is the dissolution. Bioequivalence expectation is usually bioequivalent, but the dissolution profile should be matched in all of the pH, including the pH of the medium dissolution. And some of the product also we use surfactant for the dissolution test. BCS class 3 is normally the absorption rate is based on the permeability, and the expectation is equivalent if the study power account for the variability. So this is normally the BCS class 3, we can get the good result, but be careful in terms of the number of subjects. For BCS class 4, this is absorption rate control is case by case, and the bioequivalence expectation is unpredictable. So when we discuss all of this BCS class, the most challenging one is the BCS class 2 or 4, and also BCS class 2, some of the BCS class 2 also challenging. But if you are discuss for the BCS class 1, normally we will get the good result. BCS class 3 also normally we will get the good result, but the high number of subjects. So be careful for the BCS class 2 and BCS class 4 during your development. Before we conduct the bioequivalence study, we need to be more prudent in review of your dissolution profile. And this is the gastrointestinal tract, because the bioequivalence study, the product will be we consume orally and go to our stomach. Our stomach is presented in our dissolution testing as the pH 1.2. So the first time that drug will be dissolution will be the under stomach and represent of the pH 1.2 in the dissolution testing. The second is for the duodenum, we can see here, yeah. Also for the in terms of the pH is like 4.5. Like the 4.5, we need to we need to make sure also the dissolution, but or on point 4.5, this is not really high because the area is also small, 0.09 meter square. So this is not really concern, but we should be careful in terms of the product in jejunum, because jejunum we can see here like the total of the area is 60 meter square. So maximal drug concentration also under jejunum also ileum. And the control in our in vitro will be in pH 6.8. But we will see later about the specific medium dissolution about the pH. But why we make the dissolution in pH 1.2, 4.5, and 6.8, this is the theory that support the dissolution in 3 pH. Yeah. Okay. The sampling points for the bioequivalence study, we already explained the dissolution. The sampling points that we need to conduct is 5 minutes, 10 minutes, 15 minutes is also very, very important. So do not forget to mention and to take the blood to take the sampling for the 15 minutes. This is important. And the condition, I think this is also the common information that you already know all for the number of product, 12 of the reference and 12 of the test drug. I mentioned here is we need to do the dissolution in the US FDA medium or the discriminating media. Critical points is before conducting the CDPs, you should know the BCS class and also the Tmax of the developed product. Tmax is when the concentration maximum is achieved in our blood. So we need to know these two information. Tmax also we need to know. After we know these two information, we can perform the CDP in targeted pH based on the Tmax. If the median of the Tmax is lower than 1 hour, the targeted pH in our bioequivalence study is pH 1.2, meaning that in dissolution 1.2, the dissolution should be good result. Yeah. So if your CDP result in pH 1.2 is not very good for the Tmax below 1 hour, it will be a challenge. It will be a dangerous if we continue to the bioequivalence study. So it will be good. The example of this molecule is the clopidogrel form 1. This is the target. The Tmax is below 1 hour. So it should be good for the dissolution in pH 1.2. If the median of the Tmax is 1 until 3 hours, the targeted pH is in all of pH. So these 3 pH should be good. 1.2, 4.5, and 6.8. Especially for 1.2 and 6.8, it should be very good. Otherwise, it will be dangerous if we continue to the bioequivalence study. The example of this molecule is the metformin. If the median of the Tmax is above 2 hours, the targeted pH it should be in pH 6.8. The example of this molecule rosuvastatin. For this molecule, for the example, if the molecule is not really good in pH 1.2, can we continue to the bioequivalence study? Yes. But it can be case by case, we will review. But normally we can say yes. If the pH 1.2 is not really good, we still continue. But the pH 6.8 should have the very good result of the dissolution profile. And the summary of the dissolution profile, if the product is categorized as BCS class 2 and 4, we should be prudent. So we prefer to add CDP in medium release or in simulated gastric factor. And also if the product is in extended release and also the delay release, we prefer for the extended release extend point sampling in pH 6.8. So normally we have the 60 minutes for the blood sampling, but for the extended release, we can extend the blood sampling we can extend the dissolution sampling during our CDP. We can extend like for how many hours based on the release of the molecule. For the example, like divalproix extended release, we extend the CDP until 24 hours. Because in the bioequivalence study, we also can see the Tmax of this molecule is also longer. So we need to extend in terms of the dissolution profile of this molecule, the Tmax, the point sampling in the dissolution we need to extend. And also for the delay release, we prefer to add the CDP in pH 6.5. Why? Because we need to see the start release of the product. Delay release will be released is 6.8, but we don't know when we enter into the body and then going to the gastrointestinal tract. The first point of the pH in the duodenum and jejunum is 6.5. So that's why we need to conduct another media for the dissolution is 6.5. So we perform the formula optimization based on the CDP result based on these 5 critical points. Yeah. And the result of the CDP we can see based on the F1 and F2 is if the F1, the green one, F1 is below 25% and the F2 is upper 50%, that disease will be highly likely bioequivalent. But if the F1 is like be 25 to 50 to 45%, and also the F2 is a range 40 to 50%, this is limited. So this will be case by case. Some of this product is under yellow highlight here. Is some of the product can be bioequivalence, cannot be bioequivalence, but we need to see case by case. But for in the red side here, if the F1 is range for 45 to 75%, and the F2 is below 40%, it will be dangerous if we continue to bioequivalent. So it will be risky to continue this study because the result predicted is the non-bioequivalent. So this is like the relationship between F1 and F2 in prediction of the bioequivalence study result. Okay, so this is the conclusion. The key statistical consideration is the 80 to 125% for the acceptance criteria for the conventional drug, but for the highly variable drugs, we can widen the criteria based on the intra-subject CV, and we should conduct the replicate design for the widening criteria. And also perform the formula optimization based on the CD result in the targeted pH. I still have time, Dr. Shet. Can I go through for the little bit information about Equilab?
Yeah, you can give a just brief overview of slightly overview of Equilab. So meanwhile the people is asking multiple questions in the chat, then we will continue the Q&A session. You have the right to say
this
about
is 5
something.
minutes. 5 minutes. 5 minutes.
Yeah, okay.
I will go through. Yes, Equilab, as I mentioned earlier, that Equilab we are independent CRO and WHO recognized CRO in Southeast Asia, and we established in 2003. Currently, we have completed more than 1,500 completed projects, and around 140 project is ongoing. And also we collaborate not only from Indonesia and Southeast Asia country, but we also collaborate with many sponsors from another region, including from Pakistan. We collaborate with some company from Pakistan, and also we collaborate from the other from European multinational company, from China and others. So we conduct not only for the bioequivalence study, but we also perform the clinical trials, phase 1, phase 2, and phase 3, and phase 4. Laboratory testing, we have the lab to supporting the quality control of the test of the pharma company or food company or cosmetic company. And the last, we have the dermatology testing for the claim of the product claim. So we need to do the clinical study first. And this is the countries that already accepted our report. We have been submitted our report to more than 40 countries, that including Asia countries, European, and also the Africa. We see like from European, we have now more than 8 countries that accepted our clinical study report in Europe. So if you are asking about the regulatory, yeah, we can see this slide that was already approved by many countries. And this is our accreditation from the WHO, 𝐂𝐚𝐥. We also have been inspected and also we got the accreditation. Malaysia and PRA. Malaysia and PRA, they inspect us every 3 years. The last one, they inspect us in 2024, but during the COVID-19 pandemic, they didn't inspect us. And the last, we still have the valid, the Malaysia and PRA inspection. And this is from the Badan POM. And Badan POM is Indonesian FDA, and from the national accreditation body ISO 17025 and 15189. This is also for from the national accreditation body. This is for clinical laboratory. And this is our building. We are located in the south part of Jakarta in Indonesia, and we are very strong in terms of the fixed dose combination. Fixed dose combination, we have performed many for the cardiovascular and anti-dyslipidemia drugs. So this is the combination, not only single compound or 2 compounds, but 3 compounds, 4 compounds like the telmisartan, amlodipine, rosuvastatin, yeah, and the combination. We have the experience. Endocrinology combination like the sitagliptin, metformin with the other molecules, also the combination we already performed. Anti-TB and also anti-HIV drugs. Complex dosage form, we are not only conduct the tablet and the capsule drugs, but we are having experience for the implants. Implants, we are doing the hormonal drugs, implants, subcutaneous intramuscular injection, microneedle pads, and also the suppository. So all of the complex dosage form, we already having experience conducting the study. And now we show also like many of the innovative drugs now. We also having experience conducting some of the biosimilar and also synthetic peptide, including the peptide drugs like erythropoietin. We have experience using ELISA, goselerin, and leuprolide. Also, we having experience for this is for the cancer drug, semaglutide, and tirzepatide. This is for the diabetic. We having experience, and also the hormonal drug like somatropin. We also have completed the study. So the innovative drugs currently is also very, very aggressive, and we show an Equilab anticipate, and also we have development the method for this molecule. And study in patients.
Ronald, there is a bombardment of questions in my chat box. How many slides do you have?
Just only 2 slides more.
Okay, yeah. carry on quickly.
Studies in patients, we have experience for diabetic, cancer, and tuberculosis patients. And validated molecules, we have 300 more validated molecules, including the very low limit of quantification until 0.003 nanogram per milliliter. Yeah. That's all from me. Thank you very much, Dr. Sher.
First of all, thank you, Ronald, for a very, 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 very valuable insight and knowledge. And if I say there is a bombardment of knowledge to all of us, it's very good, very knowledgeable presentation. So we have some questions from our participants. The first question is from Dr. Sidra Afzal. She asked the difference between simulation and pivotal studies and how you guys are doing the simulation.
So could you repeat the question, Dr. Shed, please?
Yeah, this is the question from Dr. Sidra Afzal. She asked difference between simulated or pivotal studies and how the simulation can be done.
Okay, simulation in terms of the statistical part, right?
Okay. So yes, means that you guys are doing the simulation statistically, plotting in recruiting the subjects, not actual. Or pivotal means you are enrolling the subjects actually, right?
Yeah. So the simulation that we perform, this is like the prediction of the pivotal study. After we perform the pilot study, that we need to do the simulation in terms of the statistical part. If this is molecule is potentially bioequivalent or not. This is the first thing about the simulation. The second thing is for the simulation, if you are asking the simulation from in vitro to in vivo correlation, for the BCS class 1 and BCS class 3, normally there is actually the simulation for the in vitro and vivo correlation, but currently we haven't seen good method for the correlation, but we can assume like the BCS class 1 and BCS class 3 has the good result for the simulation to get the good result in the pivotal.
Okay. The next question is there an authentic website or a platform from where the participants can get the idea of BCS classification?
Actually, there is no platform, but we have identified like 300 molecules which one is the BCS class 1, class 2,
Yeah, it is
class
based
3.
on experience. It is based on
Yeah.
experience and literature. Yeah, you are
Yes.
right.
Yes.
The second question is from Aisha Zahid. If we have 3 strengths, let's say 10 mg, 20 mg, and 30 mg, so can we perform the B study of middle strength, that is 20 mg? It will cover all strengths. So I think it's no, she should do the B study on the higher strength, right?
Yeah. Basically, we can do in the highest strength. We can do in the highest strength, and the lowest strength we can do the biowafer based on the CDP profile. And the requirement for the biowafer is the dissolution profile. You should have the good result.
Okay. Aisha Zahid, आपका next question जो है वो same वही है. So you can have the answer. How do regulators view B studies conducted at CROs outside the country? Are additional audits documented required?
Okay. So based on the regulators, some of the regulators like the 𝐁𝐬𝐩𝐞𝐜, we already have performed the inspection from 𝐁𝐬𝐻RA, and this accreditation that can be like a ticket to register the other countries in European. Actually, this is based on the requirement from the regulatory authority. Yeah, they need to inspect us or not. But based on our experience, like many countries in the European countries, they mention no need to inspect us because 𝐁𝐬𝐻RA already inspect us.
Okay. Asma Eric was asking bioweaver of fixed dose combination of drug. I think no. Ronald, you also have the same point of view. We need to do the bioequivalence study mandatorily on biowaivers. The next question is BCS class 3 also applicable for biowaiver in some cases? In some cases, it depends on the regulatory authorities.
Yes.
If the regulatory authority is allowing you, so you can submit the biowaiver. Otherwise, you have to do the bioequivalence. Ronald, I am just going quickly wherever I need to take your help, because I will take you on board for the answering. Can we get okay? And how? Can bioequivalence study conducted on lab scale trial batches or needs to be full scale production batch?
Yeah,
I think it's on the section.
Yeah, based on the guidance, it should be from the commercial.
You are muted. Ronald, you are muted.
Hello, hello. Can you hear me now? Can you hear me now?
Yes.
Hello.
Can you hear me now? Can you hear me now?
Yes, yes, we can hear you.
Yes, we can hear you.
Yes.
Yeah, in terms of the batch, based on the guideline, it should be produced from the commercialization batch, not from the lab scale batch. But we should have like the minimum quantity required is the 110 tablets, yeah, as the minimum requirement from the guidance.
Okay. The next question is it necessary to perform the CDP of the same bio batch with the innovator? Or we can perform the CDP before the availability of the bio batch?
Yeah, my suggestion is we need to do the CDP before bio batch, so we can have like the preliminary data, the assumption before we conduct the bio batch. We should to know the CDP prediction. So when we upscaling to the bio batch, the result will be like the same with from the lab scale CDP and the bio batch CDP. So we suggest to do it before the bio batch scale.
Okay, the next question, we only have 1 or 2 questions remaining. Can we use already performed in vitro data prior to B study, which we can perform in our lab? Or is it compulsory to perform the in vitro studies by the CRO? I think by the CRO is mandatory.
Yeah, If you already confident with your CDP, we suggest no need to conduct the CDP again in the CRO. But normally CRO will ask you the 3 pH, 1.2, 4.5, 6.8, and maybe the additional CDP will ask by the CRO. The purpose of the CRO is for the submitting to the regulatory. If you are already confident with your data, your CDP result, we suggest no need to conduct again with the CRO. Your data is sufficient.
Okay.
Okay.
I think Kashan Javed asked the question that what criteria determine whether to choose a partial or full replicate study design, and what are the key factors and rationale behind this? It depends on the inter-subject variability.
Yes.
What guidelines are used to widen the acceptance criteria? If the CV is more than 30, it is from as per the guideline of EMA. Have you conducted the pilot study or pivotal on semaglutide? Yeah,
Yes.
Ronald.
Yes.
A point of interest.
Okay. So why we choose this replicate design can be semi-replicate design 3 ways, or the 4 replicate designs, or fully replicate design is based on the intra-subject coefficient variation. If the coefficient variation, as mentioned, still below 50%, we suggest semi-replicate design 3 ways. But if the intra-subject CV is already more than 50%, then the fully replicate design is we suggested to conduct. Actually, there is no clear guidance which CV that we conduct for the semi-replicate design and which study that we do for full replicate design. But based on our experience, we can use this guidance to make which molecule for the semi-replicate and which molecule for the full replicate design. The second thing is related to the semaglutide. Yes, we have a lot of experience for the semaglutide. We already conduct the semaglutide injectables and also the microneedle pads. We're having experience. And now we are ongoing for the oral semaglutide drugs. And for the oral semaglutide drugs, we haven't completed the study. It's still ongoing. So the intra-subject CV, we cannot give you the data right now. Is it clear, Dr. Syed?
Yeah, I'm available. There is one more question. If we have a sacubitril valsartan having 2 strengths, 50 milligram and 100 milligram, so these 2 strengths are dose proportional or not, as the API is available in combination form. The sacubitril valsartan case. I think
So,
you need to be discuss in person with the formulation of the product so that we will be able to answer.
Yes.
One more question from Wajiha Jawad. What are the main challenges in accurately measuring bioavailability in humans?
Okay. The challenge is, the first thing is for the bioanalytical part, because the bioanalytical part is something that we develop the method, and also we should get the good result of the method, and also the validation. And also during the bioequivalence study, we need to measure the ISR, incurred sample reanalysis. If our incurred sample reanalysis is not good result, meaning that our result is not really robust. Our method is not really robust. So method development and validation is one of the critical point for the bioequivalence study. This is the first thing. The second thing is related to the clinical part for the subjects, because the subject is coming from others, and consumption is different, and also we need to standardize the process. And in Equilab, we conduct the study, and the subject we should stay in our site before conducting the study, and also the control of the consumption, control of the activity. This is also need to make the standardization. And this is two main challenge of the bioequivalence study. Yeah.
Okay. One more question from Syed Shazeb Shah. Which in vitro tests best correlate with in vivo performance for low solubility, high permeability drugs? I think the 𝐁𝐤𝐴 is the best in vitro correlation test. Ronald, you can also answer.
Okay, so this is BCS class. This is BCS class 2. Yeah, you mentioned BCS class 2. Yeah, for BCS class 2, it will be because BCS class 2, some of the molecules also we need to be prudent. Yeah, for the example, phenofibrate. Phenofibrate is the BCS class 2, and also the dissolution of the phenofibrate in pH 1.2, 4.5 is not cannot soluble. So the only one is the pH 6.8. Of course, the pH 6.8 we need to have the good result. So for this BCS class 2, we need to see in which pH that the product is soluble, then that in this pH that we should have the good result. And also we should correlate with the Tmax of this molecule. If the Tmax of the molecule is also we see it for the Tmax of the molecule, and we should correlate it with the pH medium dissolution. These two parameters we need to identify first, because this is case by case molecule.
Okay. Last two questions, Ronald. In two molecules combination drug, one drug is BCS class 1 and second is for BCS 2. What is the criteria of B study? Guy, please consider that for all fixed dose combination, BCS is applicable and bioequivalence is mandatorily required for all the fixed dose combination, for all the extended release products, for all the modified release dosage forms. Bioequivalence is mandatorily required.
Yeah.
I think I have asked all the question. If anything is remaining, so you guys can put your question. How to handle? Okay. One more question. How to handle dropouts in B study of drug?
Okay. So in Equilab, we already have the database is 6,000 of subjects. So we categorize also which subject is like the subjects that have a long stay with us on conducting the study with us. And for the existing subject, it's quite easy to manage the dropout cases, because we can tell them, hey, you should follow our procedure, you should follow our process and others, and it's more effective for to maintain them. But for the new subject, it's quite challenging to maintain them. So we doing extra effort to make sure that this subject is not drop out. How to handle the dropout cases? Of course, we should have like the communication regularly. This is the first thing. The second thing is related to the compensation. The compensation that we give, we should bigger in the in the end of the project. But we should get the approval from the ethics committee in terms of the management of the compensation to this subject. But these 2 points in terms of the tight communication and the compensation, it works to manage the minimum dropout cases. Yeah.
Okay. Thank you, Ronald. Thank you for answering all the questions. And I'm also very thankful to all the participants who join us today. The remaining questions which are in chat will be answered in person by us. And if someone has any query, so they can also contact us and we will be available to respond them. And also Ronald is also available. One more last request for every participant, please switch on their camera so that we can take a screenshot. I request again to all the participants, please switch on your cameras.